share with you Eric Boyd from Montana State. Eric had grown up in Iowa, moved to Montana State, and did his PhD, postdoc, and is now an assistant professor there. So today he's going to tell us about subglacial environments. Eric's got a new NASA Astrobiology Institute funded on life in rocks, and so we're really excited to hear what he has to say today. So without further ado, here's Eric.
All right, thanks, Jen. This is a great opportunity, and so as Jen mentioned, I'm completely inbred. I came to Montana State, and I decided I never wanted to leave. And so, hey, I've been able to make it happen. So I'm happy about that. Today, I'm going to talk a little bit about, touch on a variety of topics with respect to glacial geomicrobiology. And so this is work that we've been playing around with since 2007, and it's been a lot of fun. So between a rock and a cold, icy place, the mineralogical controls on the assembly of a subglacial microbiome. So before I get started, just so I make sure that I have enough time here to acknowledge all the individuals that contributed to this work. So a number of people at Montana State University, in particular postdoc Zoe Harold, Mark Skidmore, and a postdoc Trinity Hamilton, as well as a good friend of mine at Aberystwyth University, Andy Mitchell, a couple of good friends at Arizona State University, Everett Schock, Jeff Havig, Bristol University, primarily John Telling, and the University of Wisconsin, a good friend of mine, Eric Roden. And so this is work that's been supported initially by the NASA Exobiology program, and more recently, as Jen mentioned, by the NASA Astrobiology Institute. And so subglacial habitats, why should we care? Well, so commonly unknown to many of us is the fact that ice covers 11% of the terrestrial landmass. So it's a huge potential biome that many people don't even consider as a biome. However, during the Pleistocene, it's estimated that upwards of 30% of that terrestrial landmass might have been covered in ice. And during the Neoproterozoic, some models have it predicted where the whole Earth might have been covered in ice, and during what we would call so-called snowball Earth episodes. And so glaciers, as many of us know, contribute to global geochemical cycles and climate. And the question that we're really interested in is, what is the role of microbes in geochemical cycling in these environments? And what NASA is interested in particular, is how is life supported and sustained in these systems? And so we'll touch on both of these questions throughout this seminar. So are subglacial environments habitable? So here I have a picture from Yellowstone National Park. You can see this lovely filament community. This is not what we see in glacial systems. I only put this up here because we need to ask the question, what is it that life needs? So if we're going to think about a subglacial environment and ask the question whether or not there's life there, do we have all the requirements for life? So what does life need? Well, it needs electrons. It needs a flow of electrons. So follow the energy approach to astrobiology. So we need a source of reductant and oxidant. We also need a source of water and nutrients, the so-called follow the water and elements approaches to astrobiology. And then a question that I'll throw out there, because I think few people think about this with respect to whether or not an environment is habitable, and that is, what is the effect of the stability of a system on the habitability of the system? And we're not going to talk about that today. However, it's something that you should keep in mind as we're progressing through this seminar. So back in 2000, a colleague of mine, Mark Skidmore, actually published the first definitive paper showing that there's life beneath glacial ice. And so here on the left are just some light microscopy images of cells that they found in subglacial meltwaters. And then they took it a step further and they set up a couple of microcosm assays where they looked at the ability of microbial communities present in what we call basal ice. This is the black material that you can see underneath this clean glacial ice here in the panel on the top right. They simply asked the question whether or not that ice that has a lot of debris in it was capable of consuming or transforming nitrate and sulfate. And of course, from this plot in the center, you can see that they are, right? So whatever is present, whatever organisms are present in that dirty or basal ice are capable of reducing nitrate and reducing sulfate. And so this really started the whole plethora of studies, a whole load of groups of different researchers from across really Europe, China, and the United States got excited about this, started investigating what processes sustain microbes in these ecosystems. And so as we start thinking about what processes sustain microbes in ecosystems, I just kind of want to back up a bit and think about the assembly of communities, right? So what is it that drives the assembly of communities? And this is just an example taken from macrobiology, and this is the time-dependent assembly of a forest community, or a succession of a forest community. And you can think about this as a glacier retreating, right? So the glacier has retreated, time is actually moving left to right here. The glacier is retreating to the left of this, and it's leaving bare rock. So glaciers function to remove all the soil, all the sediments, all of the plant life, so on and so forth, and they leave an environment that's just bare bedrock. And so the question that we have, that we're interested in, is what's the role of this bedrock in supporting microbial communities? What effect do they have on conditioning this bedrock to facilitate establishment of more complex communities, and so on and so forth. But as we start thinking about this, it's important to also think about how glaciers work. And so this is just a diagram of an alpine glacier where we have a zone of accumulation where snowfall actually exceeds the rate of snow melt, and you get mass accumulation. The accumulation of mass causes this glacial ice to flow down gradient, and as it's flowing, it's constantly grinding the bedrock. And so what it's doing, in effect, is exposing fresh bedrock minerals that have not yet been weathered. It's also important to remember that these glaciers are oftentimes hundreds to thousands of meters thick. There is no light that penetrates to these glacier beds, and so any life that we find in these environments is supported by a chemical energy, and that chemical energy, we believe, is coming primarily from the bedrock. So we were interested in asking the question, if bedrock minerals are so important in controlling or in supporting microbial communities, then we might expect communities that develop in subglacial environments where those glaciers are overriding different bedrock types to differ. And so we sampled, with the help of all those colleagues that I pointed out on that second slide of my talk, 8 different glacial systems that differ. They differ remarkably in bedrock composition. I'm going to distill it down into 2 different bedrock types, sedimentary or carbonate systems, or crystalline type granitic systems. And so this is simply a multivariate separation of bacterial community composition. So to make sure everybody's on the same page, these are alumina tags, bacterial 16S tags, where each community from each of those 8 environments is depicted by a single spot here, a single diamond. Diamonds that cluster closely together are those communities that harbor very similar populations. Those that cluster distinctly harbor very different community compositions. And what we can see here is that there's a broad trend, or a broad clustering of communities that develop in subglacial systems that override sedimentary bedrock as compared to those that override crystalline bedrock, suggesting that bedrock composition really does influence the assembly of these communities. So we really wanted to hone in on this question. Are bedrock or are subglacial communities supported by bedrock minerals? And so to really get after this, we picked a glacier up in the Canadian Rockies. It's in Alberta. That's easily accessible. So it's about a 5 to 6 mile hike in. That's totally doable in a day. This glacier is Upper Devonian in age, so it's a carbonate system. Consists of limestones, dolomitic limestones. And importantly, for what we'll talk about today, is that the catchment bedrock contains about 1% pyrite by weight. And so again, we're thinking about the effect of bedrock mineralogy as a controlling feature of the communities that develop in these systems. So again, the Robertson Glacier sediment mineralogy, there's a lot of pyrite. So as you're walking up to this glacier, where this glacier is retreated, you'll commonly see rocks like this piece of dolomite here with huge nodules of pyrite and iron oxide rinds. We'll talk about the importance of iron oxide briefly later in this talk. If we look at the sediments themselves, so these are sediments that we sample from the ice caves that develop at the terminus of these glaciers. So this is a size fractionation, so different size fractions that you sieve these sediments. And you can see on the column on the far right, each one of those has a varying amount of pyrite, but on average, about 1% pyrite in this bedrock. If you take a close look at some of these bedrock, some of these rocks from the Robertson Glacier catchment, so this is a piece of dolomitized limestone, and what you see here, this is a big chunk of pyrite up here on the top left of this thin slice. And if we zoom in on this thin slice, you can see a bunch of different pyrite granules. And so there's distributed iron sulfides in these systems. So again, about 1% pyrite in these systems. And the question that we asked after we knew this is pyrite an important determinant of community composition? And so one of the ways, I'm not going to talk about today, that we're getting after this is by looking at the iron isotopes. So comparing iron isotopes and the soluble iron fraction, the iron oxide fraction, and in the pyrite itself as evidence for pyrite reduction and oxidation, but also using genetic and geochemical tools, which I will talk about today. So we were interested again on whether or not pyrite was an important determinant in the assembly of these subglacial communities at Robertson. And so the way we got after this question was we took about 6 different mineral types, so calcite, pyrite, hematite, magnetite, quartz, and olivine, so minerals that we knew were present in the system. We crushed these minerals, we sieved them to a uniform or near uniform particle size, we loaded them in these coupon samplers, we sterilized them, and then we took these coupons and we incubated them in these ice caves at Robertson Glacier. So again, these are the ice caves that subglacial meltwater flows out of, and so they provide a nice portal into the subglacial environment. So we stuck these coupons in this subglacial environment. We incubated them for about 6 months, brought them back to the lab, isolated the minerals, washed them, extracted DNA, quantified that DNA, and again sequenced bacterial 16S genes. And here's the result. So what you want to focus on, so this is a cluster dendrogram just describing the similarity in the communities that were recovered from these different mineral types in comparison to the Robertson Glacier sediment. So the Robertson Glacier sediment, RG sediment here, is just the sediment that was sampled adjacent to that coupon when we recovered those coupons. You can see that pyrite forms a tight cluster with the Robertson Glacier sediment as well as the Robertson Glacier water. So Robertson Glacier water is the suspended solids that are coming out from underneath the glacier, more or less. This is the material, the bedrock material that the glacier is excavating as it's moving down gradient. They also, that community also forms a cluster with the Robertson Glacier rock. Okay, and so this was a piece of catchment bedrock that we crushed and incubated in the same way as these other minerals. And so what this really suggested to us is that pyrite is a dominant control on the community composition in this system. You can also see that distinct communities developed on other minerals, so quartz and olivine, both silicate minerals, both harbored very similar communities, and hematite, magnetite, both iron oxides or mixed iron oxides, comprised a community that was very similar to each other. So again, the community that assembles on pyrite over that 6-month incubation period is the most similar to the native sediment, suggesting that iron sulfides or pyrite is a dominant control on subglacial community composition. So of course this brings up the hypothesis, or I guess drives the hypothesis, that these results must reflect the fact that iron sulfide can serve as an energy source. You could say the same thing about iron oxides. So we compared the amount of biomass, the amount of DNA, extractable DNA as a proxy for biomass that developed on these different minerals. And what you can see here is that minerals that can serve as either an electron donor or an electron acceptor, so an electron donor, hematite or a pyrite, or the Robertson Glacier rock, or an electron acceptor such as hematite or magnetite, harbored a lot more biomass than, say, things like calcite, olivine, and quartz. Okay, suggesting that the microbes that develop on these minerals are doing so, that colonize these minerals, are doing so simply because they're using them as an electron donor or acceptor. So the question that we ask are, do the communities that develop on these minerals, or that inhabit the subglacial environment in general, do they metabolize these different minerals? And so as a first cut, we went and we sequenced both ribosomal rRNA transcripts and genes from these environments. And those of us that are biologists know that when we look at the DNA fraction, we ask questions about who's there. And when we look at the RNA fraction, we can move a little bit more closely towards asking the question of who's active in the system. Okay, and so when we look at the ribosomal rRNA transcript abundance and compare that to the gene abundance, we see that not a whole lot of the community is active at any given time, or that we lose a lot of our RNA in our extraction procedure. But that the important thing here is that we actually were able to demonstrate an active population in the system. And so this was the first evidence, actually, RNA-based evidence for microbial activity in one of these subglacial environments. So we sequenced those transcripts and DNA, and so we'll take a close look here. The subglacial sediment cDNA, or the communities that come from the RNA fraction, are dominated by methanogens. We'll come back to this here in about 5 or 10 slides. We'll talk more about the methanogens. But what I really want to focus on for the next few slides are the bacterial communities, the abundance of organisms that are thought to oxidize iron, abundance of organisms that are thought to oxidize sulfur compounds, and an abundance of organisms that might be involved in the reduction of iron. And I want to try to relate this all to the role of pyrite, and in particular the oxidative dissolution of pyrite and the generation of acidity and intermediate sulfur and iron compounds. So the question that we're trying to ask, again, is iron sulfide driving the assembly of these communities? And so we'll take a look at this question in a multi-tiered fashion, both geochemistry. We've already looked at some molecular evidence suggesting that this is the case. So the first thing that we wanted to do was determine if iron sulfide is actually being oxidized. And so for the geochemists in the crowd, iron sulfide oxidation generates acidity, and we can use this acidity as a geochemical signature for iron sulfide oxidation, especially in a carbonate bedrock environment. So remember, Robertson Glacier is a carbonate bedrock environment that has a lot of calcite and a lot of dolomite. So the question is can we detect weathering products? Can we detect the dissolution of calcite and dolomite and correlate this with iron sulfide oxidation activity? And so when we look at iron sulfide oxidation, we're going to be focusing on the production of sulfate as a proxy for iron sulfide oxidation. The only mineral in the glacial bedrock catchment that has sulfur in it is iron sulfides. We see no gypsum in the environment. And so here is a plot of the concentrations of calcium plus magnesium as a function of sulfate. So if you're dissolving calcite, you're going to produce 1 mole of calcium. If you're dissolving dolomite, you're going to produce 1 mole of calcium and 1 mole of magnesium. And what you can see here is that there's this nice tight trend here. So the dissolution, or the production of calcium and magnesium from calcite and dolomite, is strongly correlated with the production of sulfate, suggesting that indeed iron sulfide oxidation is taking place in this environment, and it is being used to drive the dissolution of dolomite and calcite. So we were interested to ask the question of whether or not, so calcite's a lot easier to weather than dolomite. And so here's simply a plot of the calcium to magnesium ratio as a function of sulfate. And so when you dissolve calcite, you should produce an infinite calcium to magnesium ratio, assuming congruent dissolution. This is simply because there's no magnesium in calcite. And when you contrast that with dolomite, you should produce a 1-to-1 calcium to magnesium ratio. And what you can see here, as we, this is in the bulk meltwater coming out of the glacier. I should have mentioned that on the previous slide. Sorry about that. As we're generating more sulfate, as we're oxidizing more pyrite and generating more protons, we actually shift the weathering regime from primarily calcite dissolution to a mixed calcite dolomite dissolution. So we're really cranking away on that pyrite in the subglacial environment. We're dissolving a lot of mineral. And this is important for the microbial communities because, as we'll talk about here shortly, most of these communities are sustained by primary production driven by lithoautotrophic metabolisms. So in the case of calcite and dolomite dissolution, when you dissolve these minerals, you generate bicarbonate. And so we can look at this bicarbonate and delta 13C isotopes of this bicarbonate and compare that to what you would normally expect for respired carbon as a function of sulfate. And so as we increase the oxidation of iron sulfides, we're actually driving the DIC pool, the delta 13C of the DIC pool, more towards the DIC of the carbonates in the bedrock. And so this is interesting because now these organisms, which we believe are driving the oxidation of iron sulfide, are actually generating their own carbon source in the form of DIC produced from the weathering of this carbonate bedrock. So we'll come back around to this here in a few slides. So of course, I'm a microbiologist, and all that geochemical evidence just made me think, of course, biology is taking advantage of this pyrite oxidation, the pyrite, and they're involved in driving the oxidative dissolution of pyrite. And so we, a student of mine, set up some microcosm assays. So these are microcosms, minimal amendment. The only thing that we added to these microcosms was pyrite, and we just stuck them in the refrigerator at 4 degrees C, and we monitored sulfate production. So we did this in both autoclave killed controls and in biological controls. And of course, after 6 weeks, we start seeing production of sulfate, suggesting that biology indeed is involved in driving the dissolution of pyrite. So again, we're biologists. We want to know more. We want to know who's involved in driving the oxidation of pyrite. And again, most organisms that drive the oxidation of pyrite or of its intermediates are autotrophs. And so we sequenced rubisco or CBBL transcripts from these subglacial sediments, sequenced those, and found an abundance of organisms inferred to be involved in the oxidation of thiosulfate and iron. So thiobacillus and sulfuricella both are involved in the oxidation of thiosulfate. I think thiobacillus can also oxidize iron. Bradyrhizobium, cupriovetus, sideroxidins, iron 2 oxidation, and of course they use the energy that they gain from oxidizing these substrates to drive primary productivity, which we'll look at here shortly. So we were even more interested in the whole idea of who is driving the oxidation of pyrite, and so we went back to our cDNA or our RNA fractions obtained from these sediments, and we sequenced SOX-B transcripts. And so these are genes that are involved in the oxidation of thiosulfate. And we found a whole bunch of organisms that were consistent with what we found with our CBBL libraries. So things like sulfuricella or sideroxidins, a lot of organisms closely affiliated with thiobacillus. And this was great and interesting. And it turns out that at the same time we were doing all this work, we were setting up enrichments to try to bring one of these thiosulfate oxidizers into culture, and we got one. And so here highlighted in this pink square is the isolate that we obtained from the subglacial environment that is capable of oxidizing thiosulfate. And I'll show you some growth curves here shortly, but the point is that this organism, it's not perfectly related to the sequences that we find in the natural environment, which could be due to problems when we sequenced its genome, or it could be that we just simply didn't sequence deep enough when it came to looking at the SOX-B from the natural environment. So Thiobacillus species. So these are growth curves of that Thiobacillus isolate when it's growing on thiosulfate and oxygen. And what you can see from this graph is that you see a stoichiometric decrease of thiosulfate as you see sulfate increase. So it is driving the oxidation of thiosulfate, producing sulfate, and using the energy obtained from this reaction to drive the synthesis of cell biomass. And so these are all organisms grown with bicarbonate or CO2, so they're autotrophic organisms. And these microcosms or these growth curves were conducted at 4 degrees C. And we also did some growth curves with this organism as it's growing with nitrate. And so this, again, you can see a stoichiometric decrease in the amount of thiosulfate that you see, and the stoichiometric increase in the amount of sulfate produced. You can also see a stoichiometric decrease in the amount of nitrate and a concomitant increase in the amount of nitrite that's produced. And then once nitrate becomes limiting, they actually switch and start reducing nitrate. And in the process, they're producing N2O gas. So this is denitrification. These organisms are capable of oxidizing thiosulfate with nitrate. Now, of course, this only gets us to part of to where I wanted to be. And so Zoe, who's a very talented and ambitious postdoc, decided that she was going to grow these organisms on framboidal pyrite. So this is synthetically produced pyrite. So Eric Roden made this material for us and sent it out. And this is data that Zoe actually sent me this morning at 1:30 in the morning. She was determined to send me data to include in this talk, since we're supposed to be presenting unpublished data. So here is Thiobacillus denitrificans. It's a related species to this Thiobacillus organism. And these experiments were conducted at 15 degrees C in the presence of oxygen. And what you can see is, indeed, Thiobacillus accelerates the rate of iron sulfide oxidation. Pretty interesting that you also only see thiosulfate accumulate in the abiotic controls, and this would be consistent with a mechanism whereby Thiobacillus, the way that it's enhancing pyrite dissolution is through the consumption of thiosulfate. So it's pulling the reaction forward by consuming thiosulfate. And so, being the amazing postdoc that she is, she also did these experiments with nitrate. And so there's very little evidence for anoxic pyrite oxidation with any oxidant, let alone with nitrate. This is some data that she, again, sent me early this morning. And it's not terribly conclusive, but I think we're on the right path here. I think this organism can accelerate pyrite oxidation through denitrification activity. So this is a plot of the net change. So this is time 0 minus time 47.8 days. So the net change in the concentrations of sulfate, thiosulfate, nitrate, and nitrite. And I'd initially like to focus your attention on nitrate and nitrite. You can see nitrate is being consumed, nitrite is being produced. This would be consistent with these organisms reducing nitrate or denitrifying. We also, in this case, don't see any thiosulfate accumulate in the biotic controls. There's a lot of noise in the abiotic controls. And unfortunately, we don't see a net increase in the amount of sulfate. And we think that this is probably a problem with the IC run that Zoe ran yesterday. And so we're obviously going to repeat these analyses. But those electrons have to be coming from somewhere. We believe they're coming from pyrite. So we should see sulfate accumulate. And so this would be really nice evidence suggesting anoxic pyrite oxidation by this organism that we know is active in the subglacial environment at Robertson. Okay, so I told you we'd come back to the whole idea of DIC and the importance of these organisms generating their own DIC in these environments. So we did some microcosm assays to measure rates of primary productivity in these sediments. And so these are, again, minimally amended microcosms where we simply took Robertson Glacier sediments and overlaid them in a bicarbonate buffer and added trace 14C bicarbonate and looked for assimilation of that bicarbonate into biomass. And while many of us, especially those of us that play in the world of hot springs and hydrothermal environments, get really excited about extremely high rates of metabolic activities, we were excited from this data set because this is actually the lowest rate of DIC assimilation that's ever been measured. So 0.6 times 10 to the minus 17th moles of DIC assimilated per cell per day. And we can compare this with our runner-up, which is these half-million-year-old marine sediments, where it was measured rates of DIC assimilation were about an order of magnitude higher than what we see in Robertson Glacier environments. And so the importance here, and I don't have this data to present, we see a stoichiometric increase in the amount of sulfate during these assays, suggesting that these autotrophic organisms are using energy derived from pyrite dissolution to drive DIC assimilation. Okay, so I mentioned that we would come back to these anaerobes, to these methanogens, and so we've been talking up till now about primarily metabolisms that take place in these subglacial environments that are oxygen dependent. Okay, there's some evidence, like I just presented, that maybe pyrite oxidation can be mediated by anoxic or anaerobic processes, but primarily we've been talking about oxygen dependent processes. And the question that you might have is, how can we have these communities comprised of both aerobes and anaerobes, and not just comprised? The archaeal community is dominated by methanogens. In the bacterial community, there's a whole bunch of organisms that are inferred to reduce iron, things like rotopharax. And so the question that we were interested in asking is, how can methanogens and these iron reducers coexist with aerobes, and what kind of substrates are supporting their metabolism? And so the way that we've been thinking about this is really using a model that Andy Hodson put forward back in 2008. And here on the top we have our our glacial ice, and on the bottom we have our rock. And so here we are in the middle. We're between the rock and the icy place. And so as we move off axis, so you can think about these drainage networks developing in the subglacial environment, but as we move off axis and we move away from those drainage networks that are generally being supplied with meltwater from the surface, so oxidized meltwater, as we move off axis, we should develop a gradient in the amount of oxygen that's present. And there should be a point where the rate of oxygen diffusion into the sediments and the rate of oxygen consumption by those organisms that we just talked about, those organisms that are driving pyrite oxidation, should render that environment anoxic or hypoxic and allow or facilitate the habitation of anaerobic organisms in that environment. And so we've looked at a couple of different processes. This is just a real preliminary data set that a student of mine, Matt Urschel, sent to me, and that is that we've got a lot of genetic evidence for the presence of these rotopharax organisms. Well, he's got this rotopharax organism in culture, and he's been able to show that it can oxidize iron oxides that we know are present in the subglacial environment. So we know that there's iron reduction, the capability for iron reduction is there, but the process I really want to focus our attention on is methanogenesis, which is a strictly anoxic process. So that rotopharax organism is a facultative anaerobe. These methanogens are strict anaerobes. And so we have a bunch of different lines of evidence suggesting the presence of methanogens in these environments. So we have both 16S rRNA genes and transcripts detected that are closely affiliated with methanogens. We've detected methylcolumn reductase genes, and we've actually quantified the cofactor or coenzyme M itself in subglacial sediments, suggesting that these methanogens are active, indeed active. Measured sediment pore water methane at concentrations that are about an order of magnitude higher than what we find in the atmosphere, suggesting that these sediments themselves are a source for methane. And of course, we have enrichments of organisms that are capable of producing methane. And so when we take a closer look at the 16S rDNA gene diversity, and this is the same, we see the same thing when we look at the RNA or the DNA fraction. We see really two different phylotypes. We have what we call phylotype 2, which is one of these methanosarcinas. methanosarsinalis type organisms. That's highlighted here in yellow. Then we have this really novel methanogen that branches, kind of forms a bridge between the methanosarsinalis and the methanomicrobiales. So a distinct lineage. It's something like 82% related to its closest relative. So we don't know much about what this organism can do. Methanosarsinalis are kind of the garbage can methanogens. They can do a lot of different things. They can oxidize a lot of C1, oxidize or disproportionate C1 compounds, or things like acetate. Methanomicrobiales are generally hydrogenotrophic methanogens. And so we were really interested in understanding, one, what kind of metabolism these organisms are running, and two, what the source of the substrates that are supporting those that given metabolism. So we designed PCR primers to specifically quantify these two different phylotypes, and we'll come back to that qPCR data here in a second. I also turned to a friend of mine over at Schock who did some thermodynamic predictions, where he probably unrealistic substrate concentrations, so total acetate 5 millimolar, CO2 to hydrogen headspace of 20 to 80 volumes. And what he was able to show is at 0 degrees that autotrophic methanogenesis is actually more energetically favorable than acetoclastic methanogenesis. So the question that we had is, if this is the case, are those methanogens, the dominant phylotype of methanogens, are they running an autotrophic metabolism? And so we have these enrichments that you can see. I got busy between about 80 and 460 days and didn't return to these, but these are enrichments at 4 degrees C, or we incubated with either hydrogen, CO2, methanol, or acetate. We also have some controls in here, bromoethane sulfonate, which is a competitive inhibitor of coenzyme M. So that should knock down methanogenesis in effect, so in effect an abiotic control. And what we see here is that after 465 days of incubation, the only treatment that has a sizable amount of methane produced is that hydrogen and CO2 treatment, suggesting that these organisms indeed are operating a hydrogenotrophic methanogenic lifestyle. And so we turn back to these qPCR primers. Remember, phylotype 1 is that really novel methanogen phylotype. Phylotype 2 is that less novel methanosarsinalis phylotype. And so this is simply the abundance of those two phylotypes at time 0, so the sediments that were used to inoculate those microcosms, and at time 459 days. And what you can see is the only, I don't have error bars plotted on here, but you can trust me, the only treatment where you see a significant increase in the abundance of either of these phylotypes is in that hydrogen CO2 treatment, suggesting that this novel phylotype is indeed operating a hydrogenotrophic metabolism. You can also take and extrapolate from this that the doubling time of that organism under these conditions is well over a year, so 460 days. So long time. These experiments take a long time to perform. So this really got us asking the question, so if these are really hydrogenotropic methanogens, where's that methane coming from? And so this is work done by Peter Canovas, a graduate student in Everett Schock's group, where he's an extremely good gas chemist and was able to measure sediment, pore water, hydrogen concentrations of 200 nanomolar. And of course we have the elevated methane concentrations as well. And so the question we were interested in asking is, what's the source of this hydrogen in these systems that's apparently supporting these methanogens? And so to understand where we think the source of methane or the source of hydrogen that's supporting these methanogens is coming from, we need to return to our understanding of how glaciers work. So again, remember, glaciers flow. They have a lot of mass. They have a lot of material that gets embedded into the base of these glaciers, and they grind bedrock. So they're constantly exposing fresh mineral surfaces. And so some colleagues of ours at Bristol University, in particular John Telling, who's a research scientist, senior research scientist with Martin Tranter and Jim Wadham, performed some experiments where they took minerals, in this case quartz and calcite, and ground them to a fine powder, in the absence of oxygen, by the way. And then they exposed these minerals to H2O, and they just monitor hydrogen production. And what do they see? Well, in the case of calcite, they don't see anything. They don't see any hydrogen production. But in the case of quartz, they see a linear production of hydrogen. Okay, so again, we're mimicking bedrock comminution, we're mimicking bedrock grinding. Well, what's the mechanism that might be responsible for this hydrogen that's produced? And so what we think is going on is that you have silica that's more or less it's being sheared, and it's being sheared to the extent that it's generating radicals. Those radicals are, in essence, splitting water, producing these hydrogen radicals. Two of these hydrogen radicals combine and generate hydrogen gas. So this is the mechanism that we think might be responsible for generating the hydrogen that's supporting these hydrogen-atrophic methanogens. And so, of course, did the next experiment, which was to take rocks from each of these catchments, so Robertson's included here in blue, and perform the exact same experiment. And of course, what do we see? We see a lot of hydrogen being produced. And so we think from this data that it's really the comminution of bedrock, the exposure of fresh mineral surfaces, that's really driving the assembly of these communities. The presence of methanogens is made possible, of course, by that initial pyrite oxidation mechanism rendering these environments anoxic. So our current model for how this system works, at least with respect to the dominant players in this environment, is that iron sulfide is the predominant control on subglacial microbial community composition. So remember, we're talking about the oxidative dissolution of pyrite, oxidation of intermediate sulfur species. If any of us have ever worked with iron at circumneutral pH, you know that when you release iron 2, such as you would when you're oxidatively dissolving pyrite, it's going to rapidly hydrolyze if there's even a trace amount of oxygen around. And it's going to generate iron oxides or iron oxyhydroxides, which might support those iron reducers that we only briefly touched upon. Iron sulfide oxidation drives carbonate weathering and provides energy for autotrophy. And again, remember that carbonate weathering is also likely a source of the DIC that's supporting the carbon requirement for those autotrophs. Coexistence of aerobic iron sulfide oxidizers and anaerobic hydrogen-atrophic methanogens is made possible possibly by the fast kinetics of pyrite oxidation. Right? So the consumption of oxygen is what we're really talking about. So we think that the initial iron oxidation step in these experiments, in these environments, I guess in general, and in the experiments that we perform, is abiotic, and it's really the oxidation, the biological oxidation of the intermediate products, whether that be iron 2 or thiosulfate, and pulling the dissolution forward, the Le-Chatelier's principle, is what's driving the oxidation of pyrite. And it's also functioning to consume that residual oxygen, which in turn facilitates colonization by these strict anaerobes. Of course, finally, bedrock comminution of silicate minerals. And so I told you that Robertson Glacier and many of those other glaciers I showed you are primarily carbonate, but they're not only carbonate. There's plenty of silicate minerals present there. So bedrock comminution of silicate minerals generates hydrogen, which we think might fuel hydrogen-atrophic methanogenesis. And so our current model in diagrammatic form, so here's our glacial ice. Our flow of glacial ice is from right to left. We have our bedrock minerals. You can notice those pretty pyrite nodules embedded within matrix and perhaps a smaller component of silicate minerals in these environments. Oxygen's coming through from glacial ice melt, also through moulins and crevasses where we have surface input. That oxygen is being consumed through the oxidative dissolution of pyrite, primarily, we think, through the oxidation of sulfur intermediates. That process is used to drive primary productivity and the production of biomass. That oxygen, more of that oxygen is actually also being used to drive the heterotrophic growth of some of those organisms. So for example, that rotopharex organism is a heterotroph, and we know that there's plenty of other heterotrophs in this system. Comminution of bedrock, generating molecular hydrogen. We also have, remember, the production of bicarbonate in these systems, generating methane. Those two substrates coming together to generate methane. And so I'll just leave you with a couple of questions. glacial systems are, of course, a source of nutrients to oceans. Earth has experienced numerous large-scale glacial periods, and really, I think the question that we need to be asking ourselves is not only how are these ecosystems sustained during extended glacial ice cover periods, but also what role do these glacial systems play in governing these glacial interglacial time periods. And then, of course, as an astrobiologist, I'm very interested in the fact that there's glacial ice on Mars. Could similar processes be operating on some of these other planets? So again, with that, I will acknowledge my colleagues and take any questions that you might have.
Cool. Thank you very much, Eric. Let's see. Are there any questions to get started from anyone with inside the Hangout? Let's see. I have one question is from Jeff Gralnick. He says, "Why aren't you on Twitter?"
I'm doing it specifically to not succumb to Jen. Yeah, why am I not on Twitter? That's a good question. Everybody asks me why I'm not on Twitter, but perhaps the better question and the bridge question is I'm not on Facebook either. I refuse, man.
That's awesome. I'm not on Facebook.
I'm too busy.
Okay, so are there any questions on this? This is cool. So I'm real excited about the fact that you're not only doing community work, but you're also isolating organisms. Could you tell us a little bit about your isolation procedures? What kind of techniques you're using?
Yeah, sure. So that's kind of my mantra. Nobody gets out of my lab without learning how to grow things.
Amen.
Yeah, you know, it's interesting. I've made this claim many times that we can sequence the bejesus out of any environment that we want, and we'll maximally learn 40 to 50% of what those communities are capable of doing because we don't know enough about the physiology of these organisms. And so I'm sure this is something that Jeff Gralnick will agree with, and that is, you know, we've got to grow organisms and we've got to do it in parallel with a lot of these genetic techniques. And so how do we grow these organisms? And so we have a pretty extensive anaerobic setup for the methanogens and the anaerobes that we grow in our lab. And then, you know, the pyrite oxidation thing, it was surprising how easy that organism completely purified itself. I transferred that thiobacillus strain 4 times. That's all I did, and it purified itself. And we did those initial isolations with thiosulfate and oxygen and thiosulfate and nitrate. We came up with exactly the same organism. So that's not very technical, but it was pretty easy to get that bug in culture.
Is that probably just because of its acidophily?
I think it's because it's really damn good at oxidizing thiosulfate. So yeah. It was actually not the organism that I was hoping for. I was hoping for that sulfuricella organism, just simply because there's not much known about it. But we've been able to. We're learning a ton about this thiobacillus organism. It's not your typical thiobacillus, and so it's got a lot of the same capabilities. But you probably saw in that chart that it seems to. So it reduces nitrate, and then it makes a decision. It doesn't completely denitrify. It's reducing nitrate to nitrite, and then it makes a decision to reduce that nitrite once all the nitrate's gone. So it's a pretty interesting organism. We got a bunch of transcriptional work that we're excited to get going with to try to pin down the processes that are sustaining that organism a little bit better.
There are some questions that have come in, but just to follow up on that thought, are you planning on doing anything to test maybe the redox variation between different electron donors as to whether or not that influences the nitrification pathway or the extent of the denitrification pathway?
That's a good idea. You know, one of the things that, I hadn't thought about that. One of the things that Zoe is doing right now, you know, you can imagine that the energy gain between reducing nitrate and nitrite is substantial, and you can actually see that in the stoichiometries between those. You know, you can see a blip in the rate of thiosulfate oxidation once they switch over to reducing nitrite.
Okay.
And so these are autotrophs, and so she's actually looking at productivity of those cells when they make that switch, which I think will be pretty exciting. But it's in a sense it's looking at the exact question you asked, but from an oxidant standpoint instead of a reductant standpoint.
Cool, cool. Okay, well, to get to some questions that have come in over the internet or even in the Hangout. So Zach asked if you have any ideas as to why you don't see much of a difference between the RNA and DNA bacterial communities in Trinity's paper from 2013.
Yeah, that's actually a really good question. And so we do see a bit of a difference in the eukaryote communities. Not much, but so why do we not see much of a difference? I think that there's just a low basal level of activity in these systems, and that there's no real, there's not much of a temporal element. There's a seasonal temporal element. You have a daily diurnal difference in subglacial flow, and that has primarily to do with the input of surface water. Right, which as night comes on, you get less input of surface water in these systems because there's less melting. But I think it has to do with the fact that, so that you can compare this to a phototrophic ecosystem where you have a huge temporal element driving not only spatial niche space dimensions, but also a temporal niche dimension. I just don't think we have that to the extent that we do in more of these ecosystems that many of us are more familiar with. But no, that's something that, we try to address that in that 2013 paper if you want to read about some more of our ideas there. But good question.
Cool. Joe Moore asks, he says, cool stuff, Eric. Curious about your coupons. You used a different substrate in each coupon?
Yeah, good question. I wasn't very clear about that. We actually used, in this case, I think we had to use 2 of those coupons. So each coupon had multiple substrates, and they were separated from each other by a plug of glass wool. To minimize contamination, but yeah, I think that's probably worried about maybe there being some crosstalk between those minerals. But we tried to avoid that by separating them with glass wool. Yeah.
Nice. Okay. From Tyler Arbor on Twitter. First question: for communities that colonize minerals, are they biofilms, or what is a community larger scale morphology? And have you done any imaging of that?
Yeah, this was something that we were extremely interested in when we first got started, and then we quickly realized that we screwed up from the get-go. And so these minerals, there aren't flat surfaces to really image, and so we try to do confocal microscopy, and what we learned from confocal microscopy is that the communities that develop are very patchy, so it's not like a continuous biofilm, but where they do develop, definitely, you see a lot of EPS, you see a lot of other crap, and that's what we were trying to wash off in our rinses. I should tell you, I didn't present on any of the eukaryote data that we have from these minerals. What's really interesting is the eukaryotes on the native sediments. Maybe somebody in the audience has an idea as to why we see this. So in the native sediments, all the eukaryotes that we see are bacterivorous. They're ciliates. Yeah, mostly ciliates. They're there probably just sitting eating, the primary producers. On these minerals, specifically those minerals that can serve as an electron donor or acceptor. So pyrite, hematite, magnetite. We think, and those are also the minerals where you see the thick biofilms, like what you had suggested, develop. The eukaryote community is dominated by algae. Now, we don't think any of those algae are active. We think that, as this surface water is coming into the system, it's washing snow algae into the system, and they're just simply getting trapped in that EPS. There's such productive communities on those pure mineral phases that they're generating so much EPS that they're capable of trapping these algae. But, I'm open to any other explanation anybody might have as to why we see that. Good question.
That's really interesting with the algae. Okay, Zach has a second question here. He says, one thing I greatly enjoyed about this talk was the lack of metagenomic transcriptomic approaches. But that being said, do you have any plans in the works to use those methods to tackle ways these communities make a living?
Yeah, well, I appreciate that too, Zach. the reality is, so metagenomics, we have a bunch of metagenomic data, but like every other metagenomic data set you've seen, it's really complicated. These communities are very diverse. There's a ton of eukaryotes there. In fact, the eukaryote community is way more diverse than the bacterial and archaeal communities. So it's been hard for us to deconvolute the metagenomic data, and actually we're learning more from the more targeted approaches that we're using. Metatranscriptomics, we can't get enough RNA out of these systems. We can amplify out of a cDNA pool with PCR, but getting 25 to 50 nanograms of cDNA out of these, we can't do that. And you might ask, okay, well, why don't you do an MDA? Well, I've had such poor experience using multiple displacement amplifications that I'm not doing them anymore. so that's why there's no metatranscriptomics. So we feel like we're learning plenty from these kind of more targeted approaches at this point, at least.
Nice. Yeah. Okay. Let's see. We've got another question from Tyler. Is thiosulfate acting as a catalytic redox shuttle, i.e., like cycled but not consumed overall?
In the natural system, I don't know the answer to that. Well, so here's the deal. We don't see. I don't know. Is the honest answer? In our cultures, it's being consumed. Definitely being consumed. In the pyrite oxidation experiments, we can't measure it. It's being consumed so fast, at least in the presence of biology. In the natural environment and in those anoxic experiments, we think that there's something interesting going on where it might be serving as a shuttle, like you suggested. But we just need more data, more quality data to do some stoichiometries and whatnot to try to get after that question. But yeah, good idea.
Cool. Okay, are there any other questions? Wait 1 second here. This was really interesting, Eric. I posted a couple links to some papers from well over a decade ago that had postulated the sort of weathering as a mineral, or mineral weathering as a hydrogen generation mechanism. You're probably familiar with the Chappelle paper and the debate that was going on in Science in 1998 about that in subsurface environments. But I think it's really neat that you might have glacial generation of that same process. That's really cool.
Well, yeah. So that's actually more along the lines of our new NAI center, right? So that's more focused on serpentinizing type reactions and the generation of molecular hydrogen through the reduction of water by iron and whatnot.
Yeah.
This is, I tried really hard to generate data that would be similar to that. I was convinced for forever that there must be some iron-based mechanism to generate the hydrogen in these systems, and I could never demonstrate anything like that. Now, if you talk to somebody like Tom McCollum or Tori Holler, they just laugh their heads off and say, "Oh, you tried to do this at 4 degrees C. Of course you didn't see anything. You need to wait 1,000 or 10,000 years." But this silica base. So this is the way that we got on this track, or my colleagues got on this track, is if you look at fracture fluids that are being generated during earthquakes, and there's a lot of literature on this actually. That water that's coming up, it's bubbling, and it's bubbling out hydrogen. Yeah, and so the same process that's probably driving, it's just the shear of silicate minerals in the presence of water is driving the production of hydrogen in those types of systems, and we think the same thing's happening in these types of glacial systems.
Wow, it's just really cool. What an interesting mechanism.
Yeah, I agree.
Yeah. Let's see, we have one comment that came in for you here from Jake Beam. He said poly A tailing works really well for low RNA yields. If you're worried about that, I guess that's just a suggestion.
That's great. I'll talk to Jake about that offline maybe.
Cool. Okay, well, I think we'll go, speaking of offline, let's just go offline here. We can wrap up the talk, and then if anybody wants to unmute afterwards. But thank you, Eric, very much for a wonderful contribution to Micro Seminar.
Absolutely. Thanks a lot for everybody for attending.
Cheers.